Hello, dear students. I will continue the chapter Molecular Basis of Inheritance. In the previous video, we have studied about the chemical composition of nucleic acids, nucleosides, nucleotides, and polynucleotide chain. Let me continue. The first one is size of DNA. As we all know, DNA, the nucleic acid, is made up of monomers called as nucleotides, specifically known as deoxyribonucleotides. The size of the DNA varies from species to species. The size of the DNA is not same in all the living organisms. Normally, in prokaryotes, the size of DNA is smaller when compared to the DNA of eukaryotic cells. DNA is a long polymer of deoxyribonucleotides. 4 types of many number of deoxyribonucleotides are joined together to form a chain of nucleotides that is called as polynucleotide chain. So it is known as a polymer. So DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as the number of nucleotides. The number of nucleotides found in a polynucleotide chain represent the length of the DNA. But DNA is double-stranded. In such case, a pair of nucleotides referred to as base pairs and that represent the length of the DNA. Normally, RNA is single-stranded. So just the total number of nucleotides represent the length of the RNA. But in case of DNA, since the DNA is double-stranded, the length of DNA is measured in base pairs. And this means the size of the DNA is also the characteristic feature of an organism. Means, as I told earlier, the length of DNA is specific to the organism. So it becomes one of the characteristic of an organism. Some examples are given here. A bacteriophage known as 5X174 has 5386 nucleotides. So it is a bacteriophage. 5X174 has 5386 nucleotides in its DNA. Bacteriophage lambda has 48,502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pairs. And the haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. See, in case of human beings, the length of human DNA is 3.3 into 10 to the power 9 base pairs. But we should remember, this is the haploid DNA content. So the diploid DNA content or the diploid content of human DNA is 6.6 into 10 to the power 9 base pairs. So this is regarding the size of the DNA. The next one is discovery of DNA structure. We will study the DNA structure in detail. Before that, who are responsible for the discovery of DNA structure? Actually, the DNA structure is explained by, the model is explained by Watson and Crick. In 1953, James D. Watson and Francis H.C. Crick proposed a model of DNA that is called as double helix model of DNA. But actually, this model was prepared by the help of data collected by other scientists, namely Maurice Wilkins and Rosalind Franklin. They studied the structure of DNA by a technology called as X-ray diffraction method. So the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin were used by James D. Watson and Francis S.A. Crick to develop the model of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. So the idea of base pairing between the two strands of polynucleotide chains were supported to construct the double-stranded DNA model. And the observation of Erwin Chargaff, one more scientist, that for a double-stranded DNA helped a lot to construct the model of DNA. So what was the observation of Erwin Chargaff? The observations of Chargaff on a DNA molecule, particularly the double-stranded DNA molecule, is now known as Chargaff's rule. So his observations are, in a double-stranded DNA molecule, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. So this observation is called as Chargaff's rule. The ratios between adenine and thymine and guanine and cytosine are constant and equals 1. So these are the important data collected by Chargaff, Rosalind Franklin, as well as Maurice Wilkins. Based on these data, James D. Watson and Francis S.A. Crick developed the model of DNA that is called as double helix model of DNA. Now let me explain the structure of DNA, particularly the double helix model of DNA. Let me draw the diagram. Yes, so this is a diagram of a double helix model of DNA, the simplified diagram. I can see the two strands of DNA, and the two strands are paired together by a bond, particularly known as hydrogen bonds, and you can see some numbers and just observe the base pairing between A and T, G and C, as well as vice versa, means T and A, C and G, and just observe the number of base pairs in each segment. It should be total 5, total 10 in one complete turn, not 5, total 10 base pairs per turn. Let me explain the structure now. According to double helix model of DNA proposed by James D. Watson and Francis S.A. Crick, the DNA is made up of two polynucleotide chains, and hence the name double-stranded, and is specifically known as double helix. Then why it is named as double helix? The reason is the two polynucleotide strands are coiled in a right-handed fashion. Since they coiled in a right-handed fashion, the structure is called as helical structure, and hence it is known as double helix. As I told earlier, that strand, there are two strands, two polynucleotide strands, and these polynucleotide strands or the chains forms the backbone. But a polynucleotide chain consists three important chemical components: phosphate group, pentose sugar, and nitrogenous bases. But the backbone is specifically made up of pentose sugar and the phosphate group. And the nitrogenous bases are project inside. That means the nitrogenous bases are arranged internally and they are arranged right angle to the backbone. And in the diagram you can see the polarity. The one strand consists 5' end at one side. The another end is denoted as 3'. In another strand 3' and 5'. That means the strands are running in opposite direction. The two strands of DNA are running in opposite direction. That means one strand is running in 5' to 3' direction and another one is running in 3' to 5' direction. And this kind of arrangement is called as anti-parallel polarity. And the two strands of DNA are paired together due to the formation of hydrogen bond. And the hydrogen bonds are formed between the purines of one strand and pyrimidines of another strand, or vice versa. Means pyrimidines of one strand and purines of another strand. And again, the formation of hydrogen bond is very specific. Always the hydrogen bonds are formed between A and T as well as G and C. And there is a specificity in the number of hydrogen bonds also. Between A and T, double bonds, and between G and C, triple bonds. So, always the hydrogen bonds are formed between the purines of one strand and pyrimidines of another strand, as well as the number of hydrogen bonds is also specific. The formation of hydrogen bonds between the purines of one strand and pyrimidines of another strand, as well as vice versa, is called as complementary base pairing. So this is a unique feature of double-stranded DNA molecule, that is complementary base pairing. And the distance, the one complete turn, the length of one complete turn of DNA molecule is 34 Armstrong. 34 Armstrong means 3.4 nanometer. That is the length of one complete turn. The reason is the distance between the adjacent base pair. The distance between the adjacent base pairs. In one strand, A. In the same strand, the next nucleotide is G. In the opposite strand, the first nucleotide is T at the 3' end. The next nucleotide is C. Actually C represent cytosine, but in DNA molecule C, capital C represent deoxycytidine 5-monophosphate. Always the capital letters of nitrogenous bases in a DNA molecule represent the nucleotide, not representing one base. It represents the entire nucleotide. And the distance between the adjacent base pairs, one base pair is AT, the next base pair is GC. The distance between AT and GC is 3.4 4 Armstrong. 3.4 Armstrong means 0.34 nanometer. Such 10 base pairs are found in each turn. So 0.34 nanometer into 10 becomes 3.4 nanometer. If you measure it in Armstrong, that is the smallest unit to measure the length. The distance between the adjacent base pairs is 3.4 Armstrong into 10, that becomes 34 Armstrong. 34 Armstrong means 3.4 nanometer. Hope you understood. Now, the diameter of the DNA molecule is 20 Armstrong. 20 Armstrong means 2 nanometer. So that is about the diameter of the DNA molecule. So these are the important points regarding the structure of DNA molecule. We can also note down some other additional points. The coiling of the DNA molecule results in the formation of two grooves. The large size groove is called as major groove, and the another groove is called as minor groove. So these are the important features or points regarding the structure of double helix model of DNA. Now, the packaging of DNA molecule. Another important topic. You can expect this for 2 marks only diagram, or you can expect this for 3 marks with explanation. Even it may be asked for 5 marks. Packaging of DNA molecule. Packaging means how the DNA molecule is located within the cell. We will explain the packaging of DNA in prokaryotic cells as well as in eukaryotic cells separately. Let me explain the packaging of DNA in prokaryotic cells at the beginning. See, in prokaryotes, the DNA molecules are located in the cytoplasm. There is no nucleus in prokaryotes. There is a naked DNA that is called as nucleoid. In prokaryotes such as Escherichia coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cells. It is located in one particular region in the form of a coiled DNA, in the form of a circular DNA. The DNA, being negatively charged, is held with some proteins, but those proteins are called as non-histone protein. The histone proteins are absent in prokaryotic cells. Some additional proteins are there in the region named as nucleoid. So there is no nucleus in prokaryotes, but they are located, the DNA are located in the cytoplasm in a particular region, and that is a circular DNA, double-stranded DNA, associated with some proteins, positively charged proteins, but non-histone proteins, and that structure is called as nucleoid. The DNA in a nucleoid is organized in large loops held by proteins. It looks like large loops held by the proteins. So this is regarding the packaging of DNA in prokaryotes. But it is very special, very unique in eukaryotes. Before knowing about the structure of or packaging of DNA in eukaryotic cells, let me tell you the size of DNA in mammals, particularly in human beings. What will be the length of DNA, double helix, in a typical mammalian cell? See, it is 6.6, particularly in human beings. The length of DNA, that is a diploid DNA, in a single cell is around 6.6 into 10 to the power 9 base pairs. So this is in the length is measured in terms of base pairs. 6.6 into 10 to the power 9 base pairs. If you measure it in meters, it becomes 6.6 into 10 to the power 9 base pairs into 0.34 into 10 to the power minus 9 meter per base pairs. So it becomes approximately 2.2 meters. See, this is the length of the DNA found in a single cell. You know, the cell, human cell, is microscopic. Within that microscopic cell, ultra microscopic nucleus is there. Within that nucleus, the DNA is located, and the length of that DNA is 2.2 meter. Such a large size DNA molecule, such a large size means with respect to the length, nearly 2.2 meter length. Such a long DNA molecule is located within a very small size nucleus. So how it is possible? The reason is the length is 2.2 meter, but the diameter is very less. That is just 20 angstrom, as I told earlier. So very narrow but very lengthy DNA molecule is located within the nucleus by a special kind of packaging. So that is explained here, the packaging of a double helix in a eukaryotic cells. And that packaging is explained by a very unique model, a special model. And that model is called as nucleosome model. See, this is a diagram of a packaging of a double helix in eukaryotic cell. In eukaryotes, the organization of double helix is much more complex. There is a set of positively charged basic proteins called as histones. In prokaryotes, proteins are there, but not histone proteins. But in eukaryotes, a special kind of proteins called as histone proteins are found. And these histone proteins are positively charged. A protein acquires charge depending upon the abundance of amino acid residues with the charged side chains. The charge is because of the amino acids. Histones are rich in the basic amino acid residues lysine and arginine. So the histone proteins are mainly composed of lysine and arginine amino acids. Both the amino acid residues carry positive charges in their side chains. So both the amino acids consist positive charges. That's why the histone proteins becomes positive in sign. And again the diagram. So here the histones are organized to form a unit of 8 molecules called as octamers. Just observe the diagram, the labeling histone octamer, a large ball-like structure that is actually made up of 8 molecules of histones. 8 histone proteins are collectively to form an octamer. Octa means 8 histone proteins. The negatively charged DNA is wrapped. Wrapped means coiled. See, just observe the diagram. Around the histone octamer, the DNA is coiled. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. So, the histone octamer coiled with the DNA to form a structure called as nucleosome. A typical nucleosome, that means octamer with coiled DNA, contains nearly 200 base pairs of double helix. That means the DNA which is coiled around that octamer, the length of that DNA is around 200 base pairs. Nucleosome constitute the repeating unit of structure in the nucleus called as chromatin. Thread-like stained or colored body is seen in the nucleus. So this is a single nucleosome. But in the nucleus, many number of such nucleosomes appears like a chain-like structure. And the nucleosomes in chromatin are seen as beads on a string. Many nucleosomes are linked together in the form of a chain. So it looks like beads on a string. And that structure is viewed when it is observed under the electron microscope. The beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. Since the DNA is coiled around such a histone octamers, a large size DNA is located within a small space. And when the DNA undergoes further condensation, especially at the time of cell division, during metaphase stage, the condensed chromosomes will appear. So chromosomes are nothing but the condensed chromatin fibers. During that process, some additional proteins are also involved. The packaging of chromatin at a higher level requires additional set of proteins that collectively referred to as non-histone chromosomal proteins. So in the normal stage, histone proteins will be there for packaging or coiling. Under further condensation, during further condensation, some non-histone proteins are also involved. In a typical nucleus, some regions of chromatin is loosely packed. In typical nucleus, some regions of chromatin means thread-like structure, is loosely packed, loosely coiled, and that appears light in color while staining, and that loosely packed regions of chromatin is called as euchromatin. So euchromatin is a region with loosely coiled structure, and the chromatin that is more densely packed, very tightly coiled, and it stains dark are called as heterochromatin. So these two terms are very important. Heterochromatin was asked once in the final exam. And the euchromatin is said to be transcriptionally active chromatin. Euchromatin, which is loosely coiled, is transcriptionally active chromatin. That means that region easily undergoes transcription. Transcription means the synthesis of RNA. It will be discussed later. Whereas heterochromatin is inactive. Since it is densely coiled, that region is less active, inactive. So this is about the packaging of double helix. The remaining topics will be discussed in the next video. Thank you.